Es un gran placer para mí presentar a estos dos jóvenes que ustedes ven ahí, Carlos y Bernardo, que nos van a dar esta charla detrás del telón, ¿no? De la SoBLA. Eh, lo que he decidido presentarlo simultáneamente porque sería muy aburrido darlo uno por uno, ¿no? Entonces, vamos a ver qué pasa. Mira, ambos de ellos son latinoamericanos. Carlos es brasileño, Bernardo es chileno. Ambos estudiaron toda su formación académica hasta el doctorado en universidades latinoamericanas. Carlos lo hizo en Sao Paulo, en varias universidades de Sao Paulo, y Bernardo en un par de universidades de Chile. Se graduaron casi muy cercano. Carlos se graduó en el 2016 y Bernardo se graduó en el 2018 de doctorado. Ambos, otra vez, dedicaron su doctorado a canales iónicos. Carlos estudió la búsqueda de bloqueadores de canales iónicos y Bernardo estudió en canales iónicos no convencionales, llamémoslo así, no los clásicos de muchos años, sino más bien los nuevos con exinas, ¿no? ¿Ya? Ambos decidieron emigrar a Estados Unidos a la Universidad de Chicago. ¿Y por qué la Universidad de Chicago? Porque en la Universidad de Chicago está un grupo de latinoamericanos muy fuerte, varios latinoamericanos con muchas extensiones a Latinoamérica, es Pancho Benzanilla, Eduardo Peroso y todo el grupo de investigadores. Entonces ellos han atraído a muchos latinoamericanos y ambos emigraron a Chicago recién graduados de doctorado, o sea que han estado ahí en el laboratorio de Pancho desde el 2016, Carlos y Bernardo del 2018. Ambos han trabajado en diferentes proyectos, pero al mismo tiempo colaborando diariamente. Yo conozco muy bien ese laboratorio y es una interacción muy linda la que tienen entre Eduardo y Pancho y todos los chiquilines que vienen de Latinoamérica. Ambos ya están a punto de salir, están buscando trabajo, así que si por ahí en universidades latinoamericanas y de Estados Unidos, si saben de algo, estos dos muchachos están ready to go y nos van a presentar un trabajo que hicieron juntos, que acaban de publicar en el Biophysical Journal y es sobre estudiar la termodinámica de canales a través de cambios de temperatura muy rápido. Sin más, los dejo. Adelante, Carlos y Bernardo. No sé quién va a ir primero, pero
Ok. Ah,
vayan.
voy a cambiar a inglés rápidamente porque esta presentación va a ser en inglés. Thank you, Miguel, for your kind introduction, and thank you to the SoBLA, all the people in the SoBLA for giving us this opportunity to talk about our science, and especially, as Miguel was mentioning, this collaboration within the lab. And we're going to tell you a little bit about the story of how we were pursuing two different things and we met someplace in the middle, and then we developed these new techniques, these new tools. Okay, so the name of the paper is ion channel thermodynamics studied with temperature jumps measured at the cell membrane. As Miguel said, it was published in Biophysical Journal earlier this year. And this work in collaboration by me and Carlos with our mentor Francisco Zanilla and Ramon Latorre as a collaborators. Okay, so I don't think I really need to explain to you why do we care about ion channels, but I'm still gonna do it anyway. As you know, or as many of you might know, well, we have a big families of ion channels. Uh, they are all in certain way related evolutionarily. We have TRP channels, KB channels, sodium channels, and calcium channels. And when we think about channels, we obviously think about the function they have in our bodies. For example, when we think about neurons, we right away think about action potentials, and how sodium and potassium conduction drive the action potential. And the same way in the heart, the different families of ion channels play a role in shaping the form of the action potentials of the heart. We can also think about the light transduction in the eye. Also, ion channels plays a big role there, or other functions like during fecundation to prevent polyspermy. You also have ion channels there. And even in plants, the regulation of the gas exchange between plants in the plant's leaf, still, again, ion channels. So we can all agree that they are very important and they are worth studying, okay? So the other part of our work, it relates more with temperature, and temperature basically shapes life, okay? So temperature, like life has arisen in several parts in our planet, and you can find organisms that live at very high temperature, like in geysers or in hydrothermal vents, or at very low temperature, like in Antarctica. And temperature is actually a driving force in that evolution. So of course, also, it can give us some cues. When we have high temperature, we don't feel very well, and that is telling us that we are sick, right? And it also plays a role, well, when we, in our senses, like temperature allow us to enjoy a warm cup of tea or even a very nice cold mojito. So then the question is, for us is, well, how do we study temperature dependence on ion channels? So there has been mostly two ways to study them. The first one will be using heating of the bath or global heating. So in this case, you have a Peltier element which can transduct ion current flow into changes in temperature. So you can use that as a system to change the bath temperature, and then you can, in this case, you can put an outside there, you can record the currents, and then depending on the current that goes through the Peltier, it changes the temperature of the bath, and then you can use that type of techniques to study the temperature dependence of ion channels, like in this case, TR-PMA. Okay. Let me just move this from here so it doesn't bother our presentation. Okay, so that's the first way in which you study temperature dependence. Another way is using local heating. So in the term local heating basically means that you heat up part of the solution. In this case, it's passing current through this resistor here. The tip will heat up, and then you can put a patch pipette in front of it, and you will change the temperature depending on the current that you pass and the distance from the heater, right? So that's another way to change the temperature. And other ways is like just using infrared light to heat up the solution. And in that way, you can heat up the tip of a pipette and record the currents. And this has been used for studying the temperature gating of the TRPV1 channels. Okay, so we have these two ways, but they both have different advantages and disadvantages. So when you heat up the bath, that is in general an easy system to set up. You can buy system that can change the bath temperature. The temperature is homogeneous, so you don't really care too much where you measure the temperature of your bath, because all the bath is at the same temperature, and usually the temperature doesn't change during the experiment. But the problem with that is that it's slow, so it only allows for steady state measurements. On the other hand, the local heating techniques, well, requires usually non-conventional and custom-built equipment. So far, it's been limited to patch clamp. You also have temperature gradients in your bath, so then you need to measure the local temperature, which means that you need to have an external probe to measure the temperature. Also, it leads to temperature noise because the temperature might change a little bit during your experiment, and you have to account for that. And the position of your probe, or where you heat and where you're recording, it's critical. But of course, it allows for fast measurements, so allows to actually see the kinetics in response to temperature. So our work basically, it's trying to expand these local heating techniques to other preparation in oocytes and overcome some of the technical limitations. So for that, the first part was developing a method to generate temperature jumps in oocytes using the melanin from the oocyte. And the second part is using the intrinsic properties of the membrane, in this case it's the capacitance, as a thermometer. So we don't need an external probe to actually measure the temperature. Okay, so why capacitance? I'll give you a brief history of what the temperature dependence of capacitance. So in 1965, measurements of the impedance in the squid giant axons by Taylor showed that the capacitance of the membrane changed about 1% for each degree Celsius. And for everyone of you that might have experience with recording, usually you have a big capacitance when you record a cell or a patch. So 1% will generate a big difference in your recordings. And it's something that is all actually kind of easy to measure because it's so big, the capacitance in general. Okay, so next, in 1989, Parker identified that when you turn on a lamp in top of an oocyte and you heat it up, you generate these currents that are here that he called displacement currents because first they are always inward, and when you turn off the light, there's always outward. Okay, and this is because of the heating of the oocyte. Okay, but only in 2012 did they associated these currents due to changes in capacitance that come from the change in temperature. So here's an experiment where they basically applied and this is work from the Bessanilla lab. That's when the Bessanilla lab came in, and they actually measure the currents when you apply a pulse of light, of in this case infrared light. You heat up the water and you get this current. This is in bilayers. Okay, so they show that this has intrinsic properties of membranes. And what they did is that they actually measured the capacitance. And of course, the scales here are a little bit different. So this is when you turn on the light. As soon as you turn on the light, you increase the capacitance, okay? And then when you turn off the light, well, because of the heat dispersing, the capacitance starts to go down. So what's the reason behind this changing capacitance with temperature? So, well, what happens with temperature is that the membrane actually gets wider and thinner. Okay, so if we look at the capacitor equation, where the capacitance is proportional to the area of the capacitors and the distance between the plates, well, you can clearly understand how an increase in the area and a decrease in the thickness of the membrane will increase the capacitance. Now, how is this relationship? Well, we basically took data from measurements in bilayers of area and thickness. So, and this is basically an extrapolation of that. So if you actually calculate the area divided by the thickness of the bilayer, you find that it has this shape, but in the physiologically relevant range, it's actually kind of linear. So it's actually a linear process in those temperatures. So now the charge of the that is stored in the capacitor is equal to the capacitance times the voltage. And you will see here that then when you change the temperature, like if we follow this equation, when you change the temperature, and since this is a more or less linear relationship, you actually generate a capacitive current, okay? And since this is done using light, usually, we call these optocapacitive currents. So we're gonna talk a lot about the optocapacity effect, optocapacitive current, that is what we are referring to, okay? So, well, in the lab, people has been using these optocapacitive currents now to stimulate neurons. So what's the deal here? So if you put a nanoparticle or something that absorbs light very close to the membrane and you change the temperature, you will generate an optocapacitive current. So you can see here, this is a neuron under current clamp. If you apply a little bit of current, you will generate an action potential. But now if you illuminate this particle, you also generate an action potential. And that is because this changing capacitance generates an inward current. That inward current generates a depolarization, and that depolarization triggers the action potential. And the important thing here is that if I show in the equation before, the current that you get is proportional to the change in temperature. So the faster you change the temperature, the larger you get, you have a bigger current, so you're more efficient when you generate this depolarization, and it's better for generating the action potential. So this is where was more or less the status when I came into the lab and I was trying to study this process a little bit further. So what I did is, well, now I have another neuron under voltage clamp, and I give different like different voltages. You can see here the currents, the inward sodium current and the outward potassium current. And then in another series of experiments, I apply a laser pulse, but at the end of the voltage protocol. Now you can subtract these two recordings, and you can see that especially at depolarized voltages, there's a big current that appears, okay? And at hyperpolarizing voltages, you have the optocapacitive effect. So with these currents that appear at more depolarized potentials, we think we basically address that, well, this is just a change in the conductance through the ion channels, because when you make an IV of this, it looks very similar to an IV of the ionic currents. Now, we wanted to study this in more detail, but there's a problem when you are using this neuron system to study the effects on single conductances. And one of the problems is that, well, in a neuron, you have several ionic permeabilities, so you cannot really address or without pharmacology a specific conductance that you're studying. And I think the most important problem for us was that, well, we're not actually hitting the membrane homogeneously, because since we're just hitting a particle that is close to the membrane, but it's not covering all the membrane, you have a temperature gradient in the process. So that's a problem for us. And that's where Carlos comes in.
All right, so then at the same time, I was doing experiments using oocytes in the lab. And the idea of those experiments, we had like the shaker potassium channel, and then we're trying to label those channels. And then, after labeling, we were trying to stimulate those cells using lasers. The problem is that when I use the controls, we start to seeing this current here, as highlighted, that is essentially a heat effect on those preparations. So then we're always talking, and then we're always brainstorming, and then one day we're talking, then we said, oh, what if we combine these two things that we were doing here, and then we can now generate a temperature change using oocytes, and then using that to study the ion channels. So essentially that's what we did here. So the whole idea is that we have the oocyte, here is the cut open system in a schematics representation. Here's the melanin of the oocyte. So as we illuminate with the laser, the melanin heats up, and then it changes the temperature near the membrane. And then we can see the change in temperature here by the hallmark of the optocapacitive current. So when we see these currents, means that we're seeing change in temperature. So how we did this in reality, right? So this is a schematic representation. So then in reality, is something like this. So we have a current modulator, we have our laser, we are using a homogenizer here, and then we have the pipettes for the voltage clamp to sense the voltage here, and also the temperature to measure the temperature using a patch pipette. That we'll talk about this a little bit in details later. So the system that we are using looks like this. So we have here an example when the laser is off, and then here's focus on the dome of the oocyte. And then coming back here, we have the laser on in the dome of the oocyte. And it's important to highlight that in this preparation, the illuminated area essentially is the area where we have voltage clamp. So all the temperature change is in the area that we are clamping the membrane. And the idea of the homogenizer is that we transform a laser that is essentially a light in the shape of a circle. And if we look here, the area of the circle here and the diameter of the circle essentially is less than diameter, is essentially the same size of the dome of the oocyte. Okay, so then there was a question from the reviewers that how do we really know that that is homogeneous, the heat dispersed through the dome of the oocyte? So then for that, we had to change our system and do another experiment where we use now a thermal camera. So what we did here, essentially, we have, again, the current modulator, the laser, and now the homogenizer, but now we insert a glass slide here. And the idea of the glass slide is that it allows the light to go through. However, it doesn't allow, it allows the infrared to be reflected to the thermal camera here. And for that to be able to see the infrared coming from the oocyte, we have to cool down the glass slide using like a copper tape on ice. And if we take a look at a picture of the top part of the oocyte, of the top chamber, and then we highlight here where the oocyte is more or less sitting on, and then we can see here that the heat in this movie, that the heat develops very homogeneously. And then also we went further and then we analyzed pixel by pixel, and then we can see that in the middle they are almost the same, but as we move towards the edge, the temperature doesn't look as homogeneous as in the middle. It's just because now we have a contribution of different places that are not heating up is the same as in the middle part. But the conclusion of this experiment that essentially the heat is homogeneous, right? So then, as I mentioned before, the very early stages of those experiments, when we were doing this, is that we were using a temperature measuring pipette. And the idea of that is we calibrate this pipette by the resistance. And since the resistance changed with temperature, we would know how much the temperature was changed. And then we have here an example of the measure of the temperature in red, and it follows the square root of the time, essentially what is predicted by the model and by the optocapacitive current and the heat diffusion equations. And in the purple here is just to give an idea where when the laser is on. However, using the pipette generate two problems. So the main problem is that it turns the experiments very impractical, because every time you need to calibrate the pipette, and then we have the laser, we have another pipette for the voltage clamp. It makes everything harder. And the other thing is that we have a low spatial resolution. And what does that mean? It means that, for example, if we place the pipette not in the same place, we may have differences in the temperature. Therefore, we might be underestimating it. And then even if we try to be very, very careful and close as possible, put the pipette as close as possible, we always have like micrometers of differences, which will underestimate our measurements. So then again, the idea here is the following. So maybe we can use the capacitance, right? The capacitance, it changes linearly with the temperature in the physiological range. However, there's another thing here. How fast can we measure capacitance change? And that was the bottleneck for us. And then we had to devise a different approach to measure capacitance that Bernardo is going to talk about now.
Okay, so yeah, I just want to say that if you have any question about the methodology or what we're doing here, please feel free to ask questions.
Yeah.
I'll be happy to answer. And Carlos too.
Yeah.
Okay, so now, oh, I can bring this here. Okay, so then the question becomes, well, what's the relationship between capacitance and temperature, as Carlos said, and how we can we measure the capacitance? So for the outside membrane, we have an equivalent circuit that is basically something like this, okay? So we have the membrane capacitance, the membrane resistance, and then the series resistance between our electrodes and the membrane, right? How do we measure the capacitance in a system like that? Well, there's basically two ways. We can use a discrete way in which we basically give a voltage pulse. See, with the voltage pulse, the capacitors get charged, right? So then we have this capacitive current, and if we integrate the current, we get the charge. And from the capacitor equation, we get that the capacitance is equal to charge divided by voltage. And that's one way to measure capacitance. Now, what's the problem? The problem here is that we need to wait for the capacitor to charge. And that becomes a problem if we want to have a very fast system to measure temperature, because that would be we will need to wait like 1 millisecond at least to make every measurement. So the time resolution will not be great. Well, what's the other way to measure capacitance? Well, we can use a continuous wave. So, and this is because if we apply a sinusoidal voltage wave, you get a sinusoidal current wave. And they will have a difference between them. They will have a phase shift between the voltage and the current. And of course, the amplitude of the current will also depend on the parameters of our system, the capacitance, the membrane resistance, the series resistance. So we can use this now to measure the properties of the membrane. So in this way, in this continuous wave, when we use now, we're working in the frequency domain. So things become a little bit more complicated, okay? Because now we're talking about impedance, okay? So basically, the impedance is what in the continuous way you will call a resistance. And the impedance will be then the division between the complex voltage and the complex current. And we're gonna talk a little bit later how we get these values, okay? But the important thing is that the impedance is given by this equation in this system, okay? It might be a little bit difficult to see at first, but the important thing here is that if the multiplication between frequency, membrane capacitance, and membrane resistance squared is way greater than 1, then the imaginary part of the impedance, this part of here, will be inversely proportional to the membrane capacitance. So we just need a way to find the imaginary part of the impedance. Okay, and for the system that we're using, like for the oocytes in the cut open, we can predict that we can use frequencies of 300 or more, and we will be within this range, and the approximation will be valid. Okay. Now, how do we get the impedance? How do we get the imaginary, the real component of the voltage, current, and the impedance? So to do that, we apply what is known as the Hilbert transform. So the Hilbert transform basically transforms this sinusoidal wave into it will give us the imaginary component of this sinusoidal wave, and then you have the real component and the imaginary component. Okay? So basically, we apply this Hilbert transform, we divide the complex voltage and current, and we obtain the imaginary component of the impedance. And from that, we can actually, it's just a easy relationship to get the capacitance. And for this recording, this is basically the capacitance trace. And the red line, it's filtered at 10 kilohertz. So the nice thing of this method is we get a very good time resolution, which is even, the time resolution is higher than the actual frequency that we use to measure the capacitance, which is great for what we want to do. Because especially these temperature steps, the temperature can change widely in 1 millisecond. So we need something that is very, that we can make several measurements in time. Then, now what we need to do is to find the actual relationship between the capacitance and the temperature. And we use this system, the Hilbert transform, the imaginary component of the impedance, to obtain the capacitance. And we change the temperature of the bath to make this calibration curve. So this is basically normalized by the capacitance at room temperature. And the interesting thing is here is that we didn't find that the capacitance was actually 1% of the change with 1 degree. It's actually 1.1%. So our results are very similar to what Taylor found in more than 60 years ago. So now the nice thing is that knowing the relationship between capacitance and temperature, we can easily convert the capacitive traces or the capacitive measurements into temperature. And now we don't need external probes to measure temperature. We can just use our voltage clamp system. So let me show you how we do that. So first, we have a trace under voltage clamp where we apply the laser, and you can see it here. And this, of course, generates an optocapacitive current, okay? Now, we apply a sinusoidal voltage, and we apply the same laser. So now what you get is, well, you get the sinusoidal current plus this optocapacitive effect. So then what we can do to obtain just the sinusoidal current, we subtract. And even from here, from these raw recordings, you can already see that the amplitude of the sinusoidal is changing during the temperature jump here, this step with the laser, which is due to the change in the intrinsic properties of the membrane. So now we convert that into a capacitance, and then the changing capacitance into temperature. And we at the same time use as a validation, we use the measurement from the pipette. We call this method of measuring the temperature using capacitance as capacitance-based temperature measurement, or CTM, okay? And you can see that there's a difference between the measurements using the pipette and the capacitance. And this difference due to the distance between the membrane and the pipette. So even if we try to put the pipette very close to the membrane, we're still a couple of micrometers away. And then due to how the heat diffuses in the system, this actually generates a couple of degrees of difference. So actually this method, it's giving us the temperature just right where we want to measure, which is the membrane, which is where we're gonna do our recordings. So just as a validation, we tried using different frequencies. Even though the base capacitance may be a little bit different, when you normalize all of them, they all give about the same relative change in the capacitance and the same change in temperature. So now Carlos is going to talk you about when we bring channels into the picture.
Yeah. All right. So then bringing down channels in the picture. So thus far we just talked to you and explain how we did this technique, how we develop it, and how we measure the temperature and everything. Now the idea is that let's apply it, right? So what we do here in the lab, we have, for example, the DNA of a given channel, and then we use an in vitro transcription. We have the RNA, then the RNA we inject into the oocytes, where we have the translation, the protein expression, and here, as you can see, we have the pipette that we are using to inject the channels. Okay, the RNA, sorry. And then, okay, the first thing that we did, we tried the KIR. All right, so the KIR, essentially the voltage dependence of these channels comes from the binding of the polyamines, or for the magnesium in the inner cavity of the pore, and they have very weak voltage dependence, almost nothing. Right, and they are very important essentially to help maintaining the resting potential of those cells. So then, oh, let's do it. Let's start with a temperature jump, and then we did it, and then we found a problem. What was the problem is that in the very beginning of the currents, we have a small component here, and this component is related to capacitive currents. And then later we see the effects of the temperature. So then, oh, how can we just get the effect of the temperature jumps on the channels without the optocapacitive currents? So that we come out with an idea that we use the linear relationship, because those currents, they are linear. And then if we now take an uninjected cell, and then, for example, here, we give a laser pulse, and then the currents are going to appear here, and then we do an isochronal for all of them, we see that they are linear at any given time. So now if we do a linear fitting of those currents, and then take the currents and subtract it from the fittings, now we can obtain only the effects on the channels. That in this case here, there's no channel, so there's no effect. So therefore, we can show that we can only isolate the current. Now we can isolate the currents of interest from the optocapacitive current. However, we need to remember that sometimes we use some channels that in the voltage range they don't block. So essentially, for example, the KIR, they open all the time. And then if we need to use another voltage channels, like for example, voltage gated channels, we need to do this subtraction and the voltage range where the channel doesn't open. Or if we are using of the gating to measure gating charts, we need to use where the gating charts doesn't move anymore. So then for the KIR, what we did is the following. First, we measure the temperature jump, and then we keep the system unchangeable. We don't change anything. And then we add a blocker, in this case was barium, and then we record the currents only due to the optocapacitive current. And then later we do an offline subtraction between the laser and the optocapacitive current. And by doing so, now we get only the effects of the temperature jumps on the channel. And it's very clear to see here, for example, in these conditions, in these ionic conditions, the reversal potential is around -60, and we don't see any trace of optocapacitive current. And if we look close to this, when we look, for example, now, the ionic currents develop and the temperature jump develop for -120 or 30 millivolts. They follow the same time course. And what does this mean? It means that now, because we're increasing the temperature, the ions, they have more mobility, so therefore the current increases. So it's just a change in the single channel conductors for those voltage ranges. However, when we look at the more depolarized voltage, we see that they don't follow the time course of temperature jump and the currents. And here we can see that there's a sort of a connected process that we are going to explain later, and why we think that what's happening here is explainable by the change in the rectification of the channel. So now, if we apply isochrones here, we have like 5, for example, 1, 2, 3, 4, 5, 6. We have like different temperatures, and then we can do different IVs in only one round of experiments. We have all this much information. And as I mentioned before, in the bottom part here, in the hyperpolarizing voltage, we just have like maybe like 50%, 40% of increase that is consistent with just changing the single channel conductance. However, when we look at the depolarizing voltage, we can see that the inflection point here for the rectification is changed. And also, it becomes more current, about twice as much, which is way more than the single channel conductance would predict. So now if we take all those IVs and convert them to GVs, what we have here now, we have many GVs for different temperatures in only one single round of experiments. And then from that, if we take all the voltage where 50% of the channels here are blocked, we can get easily now, using this relationship, the delta S and the delta H of this process. And what we found that for the blocking, when the blocking happens, it reduces the entropy of the system. And we found that the entropic term of this process is very large. It's almost comparable to the TRP channels. And we hypothesize, we think that this is happening because it could be electrostatic interactions between the amines and the inner part of the cavity of the channel, or it could be some other hydrophobic interactions between the blocker and the channel. So now we show you how we can use a temperature jump. Now the next question is that can we make now from a temperature jump to a sustained temperature jump, now in this case a temperature step? And we can. How we did that? So first we apply a laser pulse where more or less we set the temperature that we want. And then after that, we modulate the laser current such as now the temperature that we set in the beginning, we're going to be oscillating that range. And then from that, we have this kind of flat temperature step here. And you can see that in less than 1.5 milliseconds, we can have a change of 9 degrees, and it maintains until the end of the recording. So now we can have a temperature step of about 9 degrees for 150 milliseconds. So then the next question, oh, how can we apply this into another channel? So then, of course, the most obvious idea would be to try the TRP channels, right? So they are thermoreceptors, so they respond to temperature. They have different set points for the temperature. They can be excited by also by chemical components, like, for example, for garlic or chili peppers and everything, like for capsaicin. And then, of course, now recently they have more attention because of the Nobel Prize for David Julius and Arden Patapudi for the discoveries of the thermomechanical transducers. And they are important for the thermal sensation and the pain sensation in our body. So perhaps what we did here, we used the TRPV1, that's the heat receptor. So as you can see here in these GVs, as the temperature increases, the GVs shift to the left, so we have more currents and more open probability. And whereas in the TRPM8 with higher temperature is the cold receptor, so it channel close or shifts the voltage relationship to the right. So first thing that we did, we tried to replicate our experiments as if we are applying a bath temperature, just to see if we have a good agreement between the two techniques, right? We expect that to be the case. So as you can see here for TRPM8, as we apply this temperature step highlighted here in the top, and then as the temperature increases, the current decreases as expected, and then we can see here better in this IV curve that the current decreases by 50%. And similarly for TRPV1, so lower temperature, higher temperature, the channel opens more, change the open probability, and the current increases about 3 to 4-fold. So it's in line what is expected for those channels. But this technique was not really meant to be like to change the bath temperature. It's actually apply this pulse of temperature at any given point of the voltage protocol. And that's why this is much more efficient and much better to use. For example, here now, highlighting here, we have the voltage protocol, and then we apply the temperature pulse in the middle. And we can see now the closure of the TRPM channels. And interesting that we see two components. One is related for the single channel conductance. Again, the same idea. We have more mobility of the ions because the temperature is increasing. But then we have a change in the channel, and then we see a change in the open probability. Similarly for TRPV1, we apply the temperature step in the middle of the voltage protocol, and then we enlist more currents, as we can see here highlighted. And again, the same process that happens with TRPM8, we can see in TRPV1, where we see a kink in the current here, where now we have the change in single channel conductance, and here we have a change in the open probability. So now using this technique, what's very nice about it, that we can do like this. We have a voltage protocol, and now we can apply the temperature step at any given time. For example, before a voltage protocol, at the same time the voltage protocol, and during the voltage protocol. You can see here. And for the TRPV1, the same idea. Before, at the same time, and during the voltage protocol. Okay, so now with this, what we can start to appreciate is the kinetics of therapy using this technique here. And what is unique about it that we are using the cut open and we can start to see more features of channels. For example, we can apply this for gating, to study the gating currents of ion channels. So it's a very robust system. And also, we don't need to label the cells, for example, with gold nanoparticle. The heat is diffuses evenly over the membrane. So with that, we have four main conclusions for this work from what we found, that the membrane capacitance can be used as to measure temperature change at the membrane. If we combine this, let's say, capacitive thermometer with the T-jumps, allows us now to get some descriptions of thermodynamics of ion channels. In KIR, the temperature relieves the blockage by polyamines. And using the T-steps in the TRPs, we start to obtain a little bit of kinetics, the response of those channels to temperature, the kinetics of this process. So Bernardo is going to give a little bit of a sneak peek here.
Yeah. So as Carlitos was saying, well, what other channels can we apply this technique on? And this is some work that we're almost, it's coming out soon to a journal next to you, hopefully. So we applied this same technique now to shaker, the KV. So you can apply, again, these temperature steps in the middle of the voltage protocol. And the interesting thing here is that, well, at very positive voltages, at 40, when all the channels open, you see similarly that the temperature and the current corresponds. And this is, as Carlos mentioned, changing single channel conductance, you have more ions moving through. But then as you go to less depolarized voltage, you start seeing effects on the kinetics of the channel. And you can see the channel that, for example, at minus 30, the channel actually starts closing, okay? So, and this we think is because, well, there is a little bit of temperature dependence in the GV of the channel. So the GV shifts a little bit to the right. And that's what makes the channel to close now. But it's nice here that now we can actually see the effect of the temperature right in the kinetics of the process. We can measure the kinetics of that process. And other interesting here is that we have found a mutant now that it acts like a heat receptor. It reminds us a little bit of TRPV1, where we change here the temperature, okay, and you get a 4 degrees change in temperature, but you get about twice as much as current, right? And this is not what you expect from a change in the single channel conductance. So that's a very interesting mutant that we're working on. And at the same time, we find a mutant that works the other way around. We find a mutant that now acts like a cold receptor. So when you heat it up, it closes. And for the whole voltage range. And you can see here is the same effect that in TRPMA, that you have first a change in the single channel conductance, but then a change in the open probability. So that's what we're working on right now, and that's this technique we believe can have a lot of applications, not only to temperature sensitive channels, but to channels in general or other membrane proteins.
So then, with that, we would like to thanks everybody for listening to us. Thanks the organizers from SoBLA for the opportunity for us to talk about our work here. Like to thanks Pinto, that our postdoc advisor, for all the mentorship through these years. And also thanks to Ramon Latorre, our closer collaborator here, our lab. We always drink some coffee and brainstorm here. And our funding agents, because for the support. Here is our GitHub. So you guys can get all the codes that we did here. And if you guys need any questions, you can please feel free to reach out either to me or to Bernardo. Thank you. Also to João, that João also introduced us to this optocapacitive world here that when we started the lab. Thank you so much for your attention, and we'd like to ask them. Thank you.
Bravo, bravo. Thank you very much.
Thank you.
So,
Felicitaciones, Carlos, Bernardo, magnífico seminario.
sí, sí, muy, muy lindo. So, ok, vamos a empezar por las damas. Andrea, tú tienes una pregunta, estás mute. Ya.
Muchas gracias. Hola Miguel, qué gusto verte. Hola Bernardo. ¿Esto es en español? Hola Carlos, ¿o lo vamos a hacer en inglés?
Comprendo un poquito, un ratito.
Ah, no, voy a hablar en inglés entonces. This was wonderful. Thank you so much. I like the duet format. That's very cool. Well, I wanted to ask two questions that are likely to impact the work that we are doing. There is Juan Pablo there as well, so maybe he can also help me out later with the interpretation of your response. I wanted to know whether you've thought of using this system in vivo in, for example, a small animal like the worm C. elegans to study channels that might sense temperature, but we don't know. So the question is sort of embedded, whether you can reveal thermosensation or thermal properties of a channel that is not described as a thermal sensor using this type of technique. Hopefully in vivo as well.
I think the main problem would be like a transducer, right?
Yeah, you will need a transducer, and then you will need a way to measure the currents, basically.
Exactly.
So,
Yeah.
but as long as you can patch it, you can measure temperature, hopefully.
Yeah, so
Yeah, these neurons are hard to patch, but this has been done.
yeah, I think the bottleneck for you would be essentially something that converts the light into heat at the membrane. So you may need to use like carbon nanotubes as Bernardo did before.
Maybe some way of labeling.
Yeah, label.
If you can make the cells darker, will be great.
Yeah.
Because then you can shine a light. I'm guessing that your organism, yeah, it's transparent or semi-transparent.
It is transparent.
So maybe we have thought about this, like maybe there's a way that you can make some cells express melanin, and in that way you can make them darker, and that will make them respond to the light. I mean, will change the temperature with the light.
Exactly.
Okay, great. Thank you.
Thank you.
Yeah, Helmut, if you'd like to ask a question.
Yeah, guys, congratulations for your work. Amazing. I can talk in Portuñol to make it easy. Two questions. One, all this experiment was done in unvitellized oocytes without vitelline membrane, or it was? Because I don't know if I can understand the part of the beginning when you do the experiment, the basic just in membrane, and most of the passive properties of the capacitant depend of the bilipid membrane, right? But what happened when you do the experiment with channels around? Was also with vitelline membrane or without it?
The vitelline membrane is still there because it helps the oocyte to stay in shape, let's say.
It would be really extremely hard to do cut open without membrane. Essentially, the cell would just and then.
Yeah, I remember I worked with connecting 26 oocytes, and sometimes when you remove the vitelline membrane and you want to do claro, the whole cell experiment is very hard. Yeah. You don't keep the shape. Okay, so what's with the vitelline membrane? So I'm gonna go a little further now. The second question is, what happened? Do you think happened in real system? For example, I don't know in neuron, but maybe in muscle, that when you have a lot of extracellular matrix also, do you think that also it helped to regulate the response of the channel to the temperature also?
Like the membrane itself?
No, to the response of the ionic channels, because at the end we are looking at the performance of the channels. And we know that, as you showed that here, certainly the capacitance, all these properties of the membrane is associated also with the protein and lipid. But what happened with extracellular matrix also? Do you think also it helped to regulate the response to temperature?
Yeah, we don't know about what extracellular matrix might be doing. we don't think it will affect the capacitance or the membrane resistance. Sometimes, whatever you have outside, what might affect is your series resistance, which is also an important, but that's more like it's a resistance for to the membrane.
Yeah.
But how
Mhm.
it will affect the temperature, diffusion, or
we don't know.
don't know.
Yeah, but there are a couple of work done in neurons using temperature change. João did. There are
Yes.
other Bernardo did too. So it's feasible to do in vivo preparation.
Yeah.
Perhaps you may need to calibrate for your system. If there is a change in the extracellular, the matrix is changing, it probably will be in the calibration curve. You probably will see that there. But we don't know.
Okay. All right. Okay. Thanks. Oh, and very good. The last part of when you this shaking mutant is now a temperature sensor. Also, that's pretty
Yeah.
good. That's how
So
biology works. You don't put tag to the channels. You just see what happens.
Yeah.
Thank you.
I don't see any more hands or in the chat room, but I have to follow Helmut a little bit comment. You were very secretive about the type of mutations you are making to make these checker channels into a cold receptor. So can you tell us, for example, at least if it is in the voltage sense on those mutations?
If they are in the S45 linker, let's say. Yes. Yeah.
All right. Okay. Thank you very much.
Very important. But I mean, there's not much secret, but we just try to keep a little bit before we send the paper out.
We want to keep you interested.
Yeah. So then you can follow up. Oh.
Yeah. All right. Sounds good. Okay, guys, if there is no more questions, thank you very much for everybody for attending this meeting. And it was really good. This dynamic duo here was fantastic. Thank you.
Thank you.
Thanks very much.
Thank you, everyone.
Thank you, everyone.
Chao. Muchas gracias.
Gracias.